Okay, can we start, ma'am?
Okay, yes.
Yes.
Do you have everyone required?
Yes, ma'am. Thank you.
So I can share my screen, right?
First, I will just give an introduction about you. So,
The presentation, yeah.
okay, just give me a
Sure.
second. Just only take 2 to 3 minutes. I don't give take much time. They are here to listen to you.
Sure,
So,
no problem.
good afternoon, ma'am, and good afternoon, everyone. My name is
Good
Sujit,
afternoon.
and we are very happy that we are hosting this Spectratix Knowledge Series, and this is the 19th edition of this series. And many of you are always been a great supportive of the initiatives that we have been taking. And Spectratix Pharma has come a long way, and also along with it, PharmaGyan Academy also has grown in terms of the volume and also the number of people that we have been able to reach. So it's a great initiative. It started as a very small initiative. Now it's slowly expanding to different regions. And you know that Spectratix Knowledge Series has become a forum wherein we try to connect and with each other and also stay updated with regard to what is happening in the industry. Because new things are happening in the industry with the advent of new technologies, new guidelines coming up. It's always good to hear from experts from the industry who come from a wide variety of sections. Continue with our tradition. Today we have with us Nidhi Jain ma'am. She's a senior quality and compliance leader with over 27 years of experience in the pharmaceutical industry. This experience has spanned across quality assurance, quality control, including oral solid dosage forms, semi-solids, nasal products, and even sterile manufacturing. She has worked with a large number of corporates, including Novartis, Glenmark, Emcure, Cipla, Vimta Labs. Uh, Her career has centered mostly around building robust quality systems in these companies. She was instrumental in driving the compliance culture and also leading global harmonization initiatives. She has led many FDA remediation programs and implemented data integrity governance and developed corporate level CAPA audit management systems. And she has also supported and led quality oversight across oral, topical, nasal, and sterile operations, ensuring that there is compliance throughout the product life cycle. She has rich leadership experience, as I mentioned, in quality control teams across laboratories, analytical laboratories, including stability programs, even release functions. And she has led many regulatory inspections and also strengthened the governance in terms of the firm itself. So I'm really happy to have you, ma'am, for this session. And it's
Thank
all
you.
over to you.
Thank you so much, Sujit, for that kind introduction. So good afternoon, everyone. Are we all ready? Yes. So today is a session for out of specification results. This is the most important topic in the industry, and if you all are in US FDA regulated or any regulated plants, so this is the most common cited topic, the investigation and especially the laboratory OOS specification. So Sujit, can I share my screen for
Yes,
the
ma'am.
presentation?
Please.
And how am I supposed to do that?
It's on the downside, the red button,
1 minute.
red arrow button.
Red arrow.
On the panel.
Just give me 1 minute. Let me check.
Just besides the audio button.
Okay, got it.
Okay.
Okay. So you all can see my screen, right, Sujit? Hello? Am I audible, Sujit?
1 second, I am not able to see the screen. Okay.
You cannot see? Can you? Yeah, Sujit.
Not yet shared, ma'am.
You cannot see yet.
Not yet.
Okay. Unable to share. We are trying to download. It's okay. It's telling unable to share.
Can you see that arrow button? Just the white one.
I can.
This side?
I did that. It is telling unable to share. I don't know why. I'll share the screen again. Entire screen. Okay.
If required, rejoin the session. It's okay. Leave the session and rejoin.
No, it's okay.
Yes.
But you can.
Yes. Now.
Yeah, you can see now.
Yes. Screen is being shared.
Okay, fine. Yeah, it's working now. Yeah, I just came out and again went in.
Yes.
So okay, sorry for that. Yeah, technical thing.
Right.
So good afternoon, everyone. So today's session, as I mentioned, is the most important out of specification results. Okay, so we will see what you all are going to go through the session, why we are going to have this discussion, what are the regulatory requirements, okay, what are the regulatory trends for this topic, what is the general flow for the investigation, understanding the various phases of OOS investigation, what are the examples of regulatory citations. I have put in few cases, and as time permits, we'll take the more cases, and then we will open for brainstorming or chatting or questions you all have. So before, like the first thing is everyone to understand that what is OS results. Now, any individual test results that does not meet established or approved specification, that is very important. Okay, you need to have an approved specification. Only after that you can tell it is out of specification result. Similarly, what is out of trend? A test results that does not follow the expected trend. Like you keep getting some marks and then suddenly one fine day, one result is less or more. That is called not in trend in comparison with either results obtained among the batch or results of the other batches, or atypical observation identified which is not obvious or questionable. So this is applicable for testing of finished product samples, stability samples, raw material sample, excipient, in process, as well as packaging material. Okay, this is a simple one. So now why? The first question you all should know, why should I do out of specs? Is it a regulatory requirement? Is it a law? Yes, it is. So 21 CFR is an FDA regulatory agency. Okay, just like we have Indian Penal Code IPC, it's a law. It's not a guidance, it's a law. What the law says, 211.92. If you pick up all the warning letters of 483s, this is the most common cited topic, 211.192. It is basically with respect to investigation. So it says any unexplained discrepancy, okay, including percentage of theoretical yield exceeding the maximum or minimum percentages established in master production control, or failure of the batch or any of its components to meet its specification shall thoroughly be investigated. Okay, please keep this in mind. It is law. Whether the batch is released or it's not released. Even if you plan not to release the batch, what FDA tells is you have to do the investigation. Okay, and a written record of the investigation has to be made and conclude. Then again, you have 820.990. This is basically for medical devices. This again says like each manufacturer has to establish and maintain procedures to control the product that does not conform to specified requirements. And this evaluation and any investigation has to be documented. Okay. Now, there are guidances which talk on, that is FDA guidance, US FDA, like all the industry follow this guidance. If it says even if a batch is rejected based on an OS result, so for out of specification, the short form in industry is OOS. Okay, the investigation is necessary to determine if the result is associated with other batches of the same product or other products. It is very important that you have to evaluate how far, whether this OS result is impacting other batches or this root cause is impacting other products also. And batch rejection doesn't mean that you don't have to do investigation. Suppose what you get an OS and you say, no, I'm not, I'm just rejecting the batch and I'm not going to do the investigation. No, you will be cited for that. Okay. Then you have Q7, good manufacturing practice guidance for API. This again tells any deviation from established procedures should be documented and explained. Okay. Then there are various 21 CFR requirements. Like for example, 211.22 clearly states that quality unit is responsible for approving or rejecting all components, a review of production records, and for approving or rejecting all procedures and specification. Okay, 211.60, 160 says general requirement discusses the establishment of specification, standards, sampling plan, and test procedures. That's the reason, why QA and quality unit is supposed to release the batches. If someone will ask us where it is written, why quality? Because the law states so. 211.160B states what is necessary for the analyst, and 211.165 outlines the reviews of specification as part of lot release. And then again, 211.165F specifies that products that fail to meet established standards and other relevant QC criteria will be rejected. It clearly says you cannot just keep it on hold and not reject. You have to take the batch disposition decision and reject the batch. Okay, and as I mentioned, there are two main guidances. This is the FDA guidance on the right side, and then you have the MHRA guidance. Now, why this? You all should know what are the types of observations the industry is getting overall, like throughout the globe, and in India specifically. If we talk, all the big pharmas involved, ignoring failing results, reporting of only passing results without justification, averaging failing and passing results together, then inadequate root cause investigation. Like this is the lowest hanging fruit. If you pick up any 483 warning letter, you will find these observations. Not setting corrective and preventive actions, then recurring problems and no root cause determined, not having an SOP for handling OSCS. Now everyone will be surprised. This was an audit observation in one of the 483 that the site did not have an SOP. Making a recommendation from OS and not following through the CAPA system, like you give some CAPA but you are not following that CAPA, no proper documentation, not expanding OS to other lots. This is again very common observation. Lack of investigation or inadequate investigations, failure investigations are not included or completed in a timely manner. Reports do not indicate if a similar OS results were reviewed and if other lots were affected. The company investigation did not extend to other batches. Okay, investigation report does not include corrective actions or unjustified conclusions. Now, as an example, where there is no scientific rationale with the lab faced with out of specification results, simply repeating the test 2 times and then claiming that the next 2 results are within acceptance, this is flawed hypothesis. Okay, this Mr. Carmelo Rosa had put in his presentation also. He's the FDA person, very well known in the industry. Without hypothesis testing, the lab cannot shift from the position that I think this is the cause to I know this is the cause. Okay, so we have to be very careful. These are the various types of observation with respect to OS. Now, what is the first step? Okay, it is very vital. If the doer, analyst notifies any anomaly at the first instance, if you're in the lab or you're working as a supervisor at present, or you're a bench chemist, or you're leading a QC team. So at the first instance, why it is very important for the analyst or the doer to notify the anomaly, what will happen? It will save the time. So you should always coach your team members, as soon as he or she feels that they have done some error while doing the analysis, they should immediately notify. This will reduce the time and resources paperwork. True, you can identify true root cause. instead of just probable root causes. Okay, you will have quick and permanent fix. Understanding the root cause provides better CAPA, and then learnings and can be shared and can prevent the errors. And clear picture, no need to invalidate merely on circumstantial data, and retesting without assignable cause can make a good product also bad product. Okay, even though your product is good, like just based on circumstantial data, you don't have root cause, it will look bad. And then improves the trust with internal as well as external customers. So invalidating an OS with an identified true root cause, assignable cause is seen very respectful and enhances the confidence of an auditor also. Okay, it builds a healthy work environment instead of blame game. Got it? So that is like what we covered is why it is required, what is an OS basic, why, what the regulation says, what are the different types of observations in the industry, and what is the first step, why, like what can be done, how it's going to help us. Now this you all can see my screen. This is an overview of the flow. Now this is taken from MHRA guidance, as I said. Okay, now US and FDA, US FDA and MHRA, there's only like the concept principle is same, but only there are few changes which I'll talk while we are discussing. So the initial part is laboratory. A person does the lab analysis. The results may be satisfactory. If not satisfactory, you all can see the red box. Okay, you have to do phase 1A investigation. What is phase 1A, 1B, we will see. And then the 1A is the preliminary investigation. What happened, just to see. You say that FIR, right? FIR. Go to the place, see what the analyst has done. Then there is 1B, that is remeasurement. Then phase 2 investigation. It has two parts: additional lab testing, hypothesis, and manufacturing investigation. Okay, and then if you get the batch disposition and CAPA has to be taken. And if again in phase 2 you don't find any cause, then it goes to phase 3 and then CAPA. This is an overall structure given in MHRA guidance. Now I'll take you through the FDA guidance. Okay, so you all can see the screen, right? So this is about the general flow. Initial identification, notification. Okay, preliminary information has to be written down. What is the batch number? What were the results? What is the STP done? Everything has to be written down. And then analyst should not discard the standard preparation and should not knowingly continue. Like if the analyst has known that, okay, shayad maine ye galti kar diya, I have done this error, you should stop there, write it and just inform. That will help a lot of things. And then immediately the training and qualification records of the analyst knowledge has to be checked, correct applicable specification was used, standard preparation has to be reviewed, instrument settings has to be seen, then aspects of chromatography, whether the current wavelength, current vials, current everything was, current setup was done, data collection, whether the calculation was done correctly, then any other cause observed, suspected during the investigation, and other batches, we have to see that. Now when you see the flow, okay, I have written that there's section, I'm going to explain the sections also in other slides. Okay, just to tell you how the flow is. The preliminary information you have to document, and then you have to see phase section A1, preliminary investigation and analyst interview. From there you may either get an obvious error, or you may get a suspect error, or you may not get anything. You will say everything is fine. In that case, you have to do the full scale investigation. Now, if you say you got an obvious error, what do you mean by an obvious error? Means when you go to the place and see that as per the STP, 20 ml flask was supposed to be used, but the person has diluted in 25 ml flask, and that's the result that there's an OS. So okay, that is an obvious error. Obvious error means you have substantial, irrefutable, substantial objective evidence. Okay, you have the evidence. And suspect means maybe he or she has done like this, maybe it is like that way. Okay, or maybe the instrument, there was a bubble, or maybe this happened. Like you just suspect, and then accordingly you go. If there's an obvious error, you have to do the reinterpretation or reanalyze. Okay, and then check the results. And if it is suspect error, then you have to do something called as remeasurement testing, that is given in the guidance also. Okay, and then as I told you, if there is no error identified, you go to full scale investigation. Now we'll see what happens if there is part 1A section, actions when obvious causes identified. As we saw over here, obvious causes identified, what can happen? Like remeasurement can be performed. when solutions are stable, or retest can be performed when solutions are not stable. Like if your sometimes solution has an expiry, right? We all are analytical people. We know like each solution when you're doing assay related substances has a solution stability. Like and if you've seen there's an obvious error. Instead of 20 ml, 25 ml or 50 ml or some other flask dilution has been done, and that's the reason the results have gone haywire. So, but the solution has also expired by the time next day you come and see. So that's when you will have to retest it with proper authorization. Otherwise, if everything is stable and you have the original stock solution and only the second dilution flask was wrong, so you just redilute it. Now, examples of obvious cases can be power outage also, incorrect instrument parameters, calculation errors, integration processing errors, okay, anything it can be. Then we can have two possibilities from that outcome. If the results meet specification and are typical for the test performed, original result shall be invalidated and reinterpreted result shall be reported. The invalidated original result shall not be averaged along with the reinterpreted. Please remember this, okay? It's very important. You cannot average the results. If the results fail or are not typical for the test performed, then you have to go to phase 2 investigation. Clear? So, okay, now let's see the 1B. We saw 1A part. That is, we have obvious cause. Now we are going to 1B part. That means it's not an obvious cause, but I think like we'll have to check whether the dilution was error or the sonication was an error. Okay, that is what we do in 1B is we remeasure with the original solutions. No new solutions can be prepared other than like if you want to prepare standard solution to compare the area of the previous standards, like the trend of the standards, you may do with authorization. Now remeasurement strategy to be planned with appropriate rationale. You have to write why you're doing revial, why you're doing resonication, why you're doing remeasurement, what all you're going to like refilter or reshake, what originally prepared solution to be utilized for the experiment. Very important. You cannot do fresh preparation test preparations here, because this is still one class phase 1 investigation. Okay, so identified causes to be simulated through experiments. Okay, suppose you say, okay, now you have done the redilution and the results are fine. That means your suspect is that the dilution initially was not correct. What might have happened? The error. So you have to do an hypothesis experiment to prove what the analyst might have done. Okay, it's not that through just remeasurement you have identified now the result is passing, so this is the error. No, now you have to prove that error, rebuild that error and show, yes, this is, if you do it like this is the error. Sometimes transient, but however transient errors, FDA guidance clearly says you cannot reproduce. Like there was an air bubble, and because of that, since I think most of you all are analytical, so you all know, okay, if there's an air bubble or pressure fluctuation happens, pressure drop happens, you cannot reproduce those errors. Okay, so you have to have proper justification, or environmental monitoring, like when you are doing the IR and there was like a lot of humidity and then you get a peak for the moisture, and that's how you say IR results are not matching to the standard. Okay, so you all got this. I'll explain the flow. Okay, what you need to do is investigation testing. You can do simulation to confirm or do the suspect cause. Okay, when the suspect cause cannot be reproduced, like transient error, as I told you, like it can be performed with precautions and results can be interpreted. Now you have to evaluate your investigational testing. Either the suspect cause is confirmed or it is not confirmed. If it is not confirmed, then you have to go for full-scale lab investigation. Then if it is confirmed, then you'll have to propose for a reanalysis plan. Okay, a retest plan has to be defined prior in your procedures, like how your retest you will be doing, and then do the retest, evaluate those results, take CAPA that these errors will not be repeated, and then evaluate and take the batch disposition decision. Okay, then now I'm explaining like what we saw in the flow part 1B, when obvious cause is not identified but a suspect cause is identified. As we saw 1A obvious cause, 1B suspect cause, and like what will happen in this case where cause is not obvious but it is suspect, lab error or instrument malfunction can be tested with the same test preparation as I mentioned regarding what might have happened. Okay, this is only like your thinking, this might have happened. It can be done either by simulation to confirm the suspect cause. When the suspect cause cannot be reproduced, like transient malfunction, environment excursion, in such case remeasurement can be performed with all precaution, and results can be interpreted, like you can repeat in such cases. Now, investigational testing in this case should not be used to replace the originally suspected analytical result. This is only an investigation testing. Okay, here you may get passing results, but it's not that you will start reporting these results. No, now you still have to confirm the root cause through hypothesis and then do a retest. Okay. So what does 1B? Study done to ascertain the suspect cause outcome. Now, option 1, suspect cause is not confirmed. In that case, you have to proceed to phase 2 investigation. If it is confirmed, now you come to know that analyst has done a dilution error, because when I'm rediluting the original solution, results are okay. Okay, that is called suspect cause. And then you do a hypothesis that if the pipette was taken, whether the full drain pipette or bulk pipette was used. If assignable cause is identified during investigation testing, then document it with rational identified cause, arrange to correct that cause, and then arrange the retesting strategy, whether you want to do in triplicate, single analyst, or in duplicate and 6 replicates. Okay. Impact of assignable cause on past and future batches and other products, like what type of cause you had identified. Like if you try to identify that, okay, this the person had used incorrect pipette, so whether his past analysis also he has used such pipettes, or for other analysis what he does, whether he uses such dilution error, you have to do the trend and basically expand that root cause horizontally. Okay, must be assessed. Got it? And then if results meet the specification and are typical, conclude the investigation and summarize the assignable cause. Trend analysis, impact to past and future batches, and corrective and preventive action. And invalidate the original results and document the batch disposition. Very important. So you got the error and you will document the batch disposition after doing the retesting. Now that was phase 1 investigation. So FDA says that if in phase 1 investigation you don't identify anything, you will have to do a full scale OOS investigation, that is called as full scale investigation. Now, when all it can be initiated? It could be initiated where in part 1A, when we saw when obvious cause or suspect cause is not identified, or after actions where obvious cause was identified and you had repeated the test, but again you got an OOS. In that case also you will have to do a full scale investigation, either that time, or in when 1B, when the suspect cause was not confirmed, that time you will have to do the full scale investigation. Now, full scale investigation has 2 parts, phase 2A and phase 2B. Like 2A is manufacturing investigation, where you have to check everything. What was the process? What were the in-process parameters? Whether there was any breakdown during the test, like manufacturing, or there were environmental excursion, whether there was issue in the sampling procedure, the materials used, everything you'll have to see in manufacturing. And phase 2B is detailed lab investigation, which covers method validation, procedure, robustness, complexity, testing instrument, and hypothesis testing. Okay, got it? So this is phase 2 investigation, phase 2A workflow. The manufacturing investigation you need to have checkpoints to be well defined, what all has to be reviewed. Okay, so you have to check to identify potential vulnerabilities in the manufacturing, like where all it can go wrong, everything. You can use your root cause analysis tool like FMEA, failure mode analysis, 6M with YY combination. Okay, any other tools as defined in your root cause analysis. If the probable cause are attributed to human error, again it has to be done through your detailed checklist, like what happened in human error, because human error is also categorized, whether it's an intentional or unintentional, what has happened. Okay, so when you do manufacturing investigation, your cause is identified, you do the impact assessment. Just excuse me. Yeah, you do the impact assessment, conclusion, containment, and take CAPA, and then take the batch disposition decision. Okay, so if the cause is not identified, you have to conclude phase 2A. Got it? So these are the various, like, points for phase 2A, like what all things you could do. You could review the batch records, BPR records, environmental condition, as I said, cleaning records, any breakdown, how the batches were manufactured, whether it was in campaign, how the other batches behaved, the raw materials used, the sampling procedures used, And for blend uniformity and blend uniformity of dosage, you have to review the sampling procedures, comparison of processing parameters with the past validation batches. Everything you'll have to do in manufacturing. So manufacturing is a very detailed investigation always. So what happens in manufacturing, as I told you in the workflow, you may get the cause identified, no assignable cause. Then you have to go to the lab investigation and do a full hypothesis testing. Or if assignable cause is identified in manufacturing, then you will have to see the impact of assignable cause on the past batches. It is very important. Okay, the details of impacted batches shall be immediately escalated to initiate appropriate market action. Like, because it's a manufacturing error, and that means that is impacting the past batches which is released in market. So of course you'll have to take some market action. Okay, and if the manufacturing investigation confirms the cause, result as process related or laboratory investigations can be terminated. Like if you come to know, okay, there was a manufacturing error, and now this batches will sure shot give OS investigation. No need to continue with the lab investigation. You can terminate that and take the batch disposition decision. Okay, during the review of other batches that might have been released, if a deviation is or identified which was not detected during initial review, that also has to be investigated. Got it? Now, what is full-scale lab investigation? It's very important to know. Now, you saw that it is obvious error 1A, then you do phase 1B, remeasurement, then you come to phase 2A, manufacturing investigation. Now, this is full-scale lab investigation. That in lab also, like when you did the preliminary assessment and remeasurement, you didn't find anything. That means now you'll have to sit down and check everything. Okay, how your lab AMB, AMT data, how is your method robustness, how is your past history of the performance of the method, whether the sampling procedures in lab were adequate, whether about the history, whether you have seen many failures with respect to this test and this method. Okay, and based on your hypothesis, whether it requires some specific technique. Okay, all these you need to design hypothesis, like what you want to, like, where you want to see the vulnerability in the lab, what could have gone wrong. And this plan, you must describe the details, what all hypothesis you will do, how many samples you will test, and what will be my acceptance criteria. All those things you will have to define. So in assignable cause, once you do that and do everything, QA will authorize that. And then assignable cause may be identified. If investigations identify assignable cause, retesting has to be done and batch disposition has to be documented. If assignable cause is again not identified, okay, and again you get OS, keep getting OS results in this hypothesis also, then you cannot do any further testing. You will have to stop and take the batch disposition decision. Okay, now if investigation do not identify assignable cause, but during hypothesis, but you don't get an assignable cause, what was the cause, but now the results are passing, but you have not confirmed the cause also. In such case, what FDA is telling, you could, like, a comprehensive laboratory investigation fails to reveal any lab error. Okay, retesting can be done. Review the events of production. Okay, whether there was no error in production. Review of manufacturing process and product history demonstrates that the product is robust. There are no market complaints. There are no validation failures. All investigation testing does not confirm to the OS. Like whatever remeasurement you did, everything was passing, but still you have not confirmed the root cause. In such cases, only in absence of a confirmed root cause, you could do a retesting, provided you have a robust product history. And this was once in a blue moon case, that you have not identified the cause also, but like all other investigation results are passing. Okay, in such case you could do the retesting. If retest results do not meet acceptance criteria, then decision of batch disposition, that is rejection, has to be documented. You cannot keep retesting again and again. Okay, suppose now you have documented the batch disposition decision and root cause is not identified. So what FDA says very clearly, even the MHRA, that you could, continue to phase 3 investigation and do your plan to identify the root cause. Phase 3 may cover manufacturing or laboratory or both for investigational purpose only. You cannot reverse your decision. In phase 3, if you say you come and identify the cause, now I want to release that batch which I had rejected. No, you cannot do that now. Okay, investigation in phase 3 shall be done through an approved investigation plan. Any retesting results at this stage will not be used to reverse the batch rejection decision. Okay, so there are timelines you have to define. Uh, any OS investigation should be initiated within 1 working day of discovery, and the initial preliminary investigation, whether it's a lab error or not, should be completed in 3 days. Because if it's a commercial stability batch, a batch which is in market and you have observed OS, and within 3 days you do not invalidate it, in that case you will have to initiate a field alert report, that is notification to the FDA that we have observed an OS investigation. Okay. Then retesting, like as I explained, you could do the when all you could do the retesting when the phase 1A, 1B or 2B, when you have identified the root cause, okay, or your solutions were not stable. Got it? These retesting plan, your SOPs need to be very thorough and very thoroughly define how many retest replicates and how many analysts would be doing the retest for me and what will be my criteria. And this has to be approved by QA before you start your retesting. It has to be authorized by QA, as I told you, and you have to use your validated sampling plans. If assignable cause is identified and retest results meet specification, original results shall be invalidated and retest results shall be reported. Very clear. Okay. In case where investigation does not reveal a cause for the OS test results, but results obtained during investigation, retest, resampling do not confirm, a full consideration shall be given to the OS results. Of course, you cannot still say that no, I'm going to release the batch. Okay. Now resampling shall be performed in case where sample is exhausted or it is deteriorated, and it has to be prior authorization from QA. Like FDA is very clear. Next topic is on averaging. Like this directly say use of replicates to arrive single reportable results and specific number of replicates should be specified in the written approved test method. Like maybe there are some methods where they say do it in 3 and then report the average, or do it in 2 sample preparations. All those number of replicates have to be well defined. Only then that averaging can be taken. Acceptance limits of that variability among the replicates also has to be well defined in your procedures. Okay. If acceptance limits for replicate variability are not met, the result should not be used. So, and shall not be done, averaging shall not be done as it may hide the variability. The final reportable value will be the average of individually passing results. Got it? Now, inappropriate use of averaging is reliance on averaging has disadvantages of hiding the variability. Like if you're doing 6 replicates and one is very low, one is very high, but the average is okay, so you will hide the variability, and then going forward one of the tablet may fail also. It may happen that way. It is inappropriate when the intent is to measure variability. Got it? So OS and inspect result should never be averaged. Again, that's very common. You cannot average a failing result and passing result. Then comes to the CAPA. The trend analysis must be performed to determine the frequency of occurrence of OS, like how many times you have observed this. Okay, whether it's repeating every 3 months, every time I'm doing the analysis, this batch, this product, and this method is giving me issue. In such case, OS taking a CAPA is very important. Okay, and historical review, generally the industry follows this. You check the past 24 months, OS in going back and checking. By 24 months, many people will ask, because most of our uh, product validity is 24 months. So this trend analysis must be performed by specific test failure, like what is the test that failed, what is the product, which instrument. Analyst also you have to trend. Okay, like person X keeps doing mistake every time, so you have to check whether this person was involved in so many analysts. That means he has to be retrained or disqualified and requalified. On job training has to be given. All those things has to be done. If the same OS is reoccurring and corrective action has been implemented, then assessment of effectiveness has to be done. Okay. So if the OS results can be attributed to clear assignable cause, the OS can be invalidated. If investigation confirms OS results, the batch disposition should be rejected according to 211.192. The investigation should be extended to other batches affected. Further investigation into root cause shall be done, and corrective and preventive actions has to be taken. If it is unconclusive, if investigation reveals no cause and does not conform to the results, OS results should be considered in any batch decision. Okay. And infrequently, a very thorough investigation may produce information that shows that the source of OS was a cause unrelated to manufacturing operation. What happens, what FDA sees is throughout the industry, many times people fail to do manufacturing investigation, and they thoroughly keep going round and round in lab itself. So any decision to release the batch despite of initial OS that has not been invalidated should come after full investigation and has shown that OS results does not reflect the true quality of the batch. While making such decision, quality has to be very careful and always err on the side of caution. Even the guidance says that. Okay. OS results in API possibility that API can be reprocessed or reworked on the basis of defined validated processes. Got it? That was in short, the complete workflow for the OS. We saw the definition, we saw what are the regulatory requirements, then we saw what are the types of observation, what are the various phases, phase 1A, 1B, phase 2A, 2B, and how the retesting, averaging, what is the importance. Now, there are some examples of 483s. I hope everyone knows what is a 483. When FDA comes to your site, does an inspection and finds objectionable items, they give you an observation that's called as a 483. Now, if you see this 483, it says there's a failure to thoroughly review your unexplained discrepancy and failure the batch of any of its components to meet any of its specification, whether or not the batch has been distributed. They are giving this trend number of investigation, what is deemed invalid, how much percentage was your invalidated OS results. So what they're telling is you invalidated initial results who retested and reported average results of replicate test. Okay, this is what they're telling. Evaporated sample solvent was identified as probable cause of the OS result. However, you did not take appropriate corrective and preventive action to ensure that evaporation of sample solvent to which attributed failure could not affect other analytical work in your lab. So this was the 483. Eventually, that site got a warning letter. So why I'm giving this is, see on the left hand side, the type of request FDA will tell you in the warning letter is, you'll time consuming also, and it's not very easy. This is a typical type of example. If you get investigation and if your response is inadequate, they find this is the type of request generally they will give for investigation that conclusively establish lab error. Ensure that other lab methods vulnerable to the same or similar root cause are identified for remediation. Any OS found by the review to have an inconclusive or no root cause identified in lab. Include a thorough review of the production, records. They're telling if you don't identify anything in lab, review the production, manufacturing steps, suitability of equipment, variability of raw material, process, everything. Okay, and analysis of overall trends in your operation, a comprehensive and independent assessment of your system of investigation, your CAPA, how the CAPA should be included, the competencies, everything they will ask you to do. And this is very, very common. Okay, if you pick up any warning letter, you can see that. So now, Sujit, we have time, right? Sufficient?
Yes, ma'am. We can.
Yeah, so we'll take examples, okay?
Yes.
So, yeah. So let's see some examples. I have not revealed any product names. These are hypothetical examples. For example, now you get OS results in a finished product batch, okay, during stability testing. There were 6 batches analyzed in the same sequence, and one batch was observed and OS was observed only in one batch. Now phase 1A, okay, we did not observe any lab error in phase 1A. So in 1B, then we go to 1B, suspect cause. No suspect cause was proposed, but remeasurement was done directly. You are not proposing, not writing what is the suspect cause, directly doing some remeasurement to rule out instrument malfunction. Okay, but we are not defining, nothing is defined. What precaution we are going to take, whether I'm going to purge the injector, whether I'm going to remove some bubbles, what I'm going to do. Re-injection of the same standard was also done from the same vial. The areas were not comparable with original preparation, and hence standard error was confirmed. Now, what was the drawback over here? Fresh vial should have been filled from original preparation and injected also. Writing analytical error in standard preparation is unclear. As result, we cannot implement any CAPA. Okay, now lab investigation was done, and based on phase 1B was hypothesized like there is a standard preparation error. Like what is that error? We don't know. They just, some people will write there's an analytical error, but what is that analytical error was unclear. So we have to be very clear in when we are writing all these things. So in case of standard error, because they are saying standard error, that means it will impact the other 5 batches which I have analyzed, right? That so all batches will have to be retested in such cases because we are telling it's a standard error. Okay. So next example we'll take is analyst documentation. Differences in AMV, and AMV is analytical method validation and standard test procedure. Now there was a case of OS results obtained in assay of cream or ointment during stability testing. Okay, you were doing a stability testing and you observed an OS result. So phase 1A, again there was no obvious error, but the supervisor in such cases, what is required is supervisor should know when he is reviewing the phase 1A that this is an ointment or this is a nasal spray, what are the critical points for that method. Like if it's an ointment, whether it's like whether it was warmed properly and then the investigation was done, the solution was very critical steps should be, in short, critical steps should be known by the supervisor. Okay, it should not be a checklist based exercise. Okay, you have done, yes, analyst says yes, and supervisor says yes, yes, yes. Supervisor should be having that much knowledge also to ask the right questions and then document it, whether you have done it properly. Since it's a cream and ointment, whether it's stuck on the flask, whether you had warmed the solution properly so that it melts. Okay, in this case suspect cause like remeasurement was done, sample was reheated and injected, and the assay increased by 25% over here. So the cause was confirmed that inappropriate heating and shaking was done. Analyst had mentioned and documented this step of heating and shaking though. So initially when a supervisor had asked him, he had told yes, he had heated. But again when it was reheated and then reshaken, the results increased by 25%. Okay, now what should have been done? What precautions were done when it was reheated and injected? Nothing was mentioned. All those things should be clear. Phase 2 manufacturing, there was no error identified. Now hypothesis was planned, experiment was done as per STP and AMV and without shaking as per STP. It was that way. This was the hypothesis experiment, like they did as per the standard test procedure, what was given in the AMV as well as without shaking. Now, STP and AMV method, when we reviewed, they had differences. What AMV said is heat the sample on boiling water bath till the ointment completely melts. Whereas the standard test procedure mentioned heat the sample on boiling water bath at 60 degree for 30 minutes. So there were differences over here. The AMV does not mention any time limit for heating, and boiling water bath cannot be like, it can be either a hot water bath that maintained at 60 degree. You cannot have a boiling water bath at 60 degree. Boiling water bath will always be 100 degrees, right? So STP requires heating on boiling water bath 30 minutes with intermittent shaking. So AMV did not have those parts. There were differences. The hypothesis was unclear. Like if sample was heated at 60 degrees or on boiling water bath. What was done was not documented in the hypothesis plan. Okay, whether it was shaken, who asked them to shake or not shake, nothing was written. So why I'm telling these examples, because these are live examples which we have handled. Okay, so all these gaps we have seen in within investigation. No experiment to evaluate the differences, like nothing was discussed on the differences in AMV and STP. Heat the sample on boiling water bath at 60 degree for 30 minutes and heat on boiling water bath were the same. Nothing was mentioned. How the previous samples were analyzed till date, nothing was. There is no sound scientific rationale for the OS results. Simply we tested the fresh, claiming acceptability if the next 2 results are within the acceptance criteria. Now this is a flawed hypothesis. Okay, please remember this. Because we shift from the position, as I told you, like I think this was the error to I know this is the error. हाँ इसने ठीक से shake नहीं किया है, heat नहीं किया है, चलो this is the error. It should not happen. You should do thorough investigation, compare everything properly, what has happened, what not. So that's the reason the supervisor also has to be equally smart enough. So now next is an example where analyst will changes the statement after hypothesis testing. Okay. Now there was a case where OS results obtained and small droplet size distribution. This is a test for nasal spray during stability testing. Now, during phase 1A, nothing was seen. As I said, the analyst supervisor should have sufficient knowledge to ask the right questions and review, because he should know which type of test will be critical. Now, 1B remeasurement was done with the original solution. OS reoccurred. Malfunction was ruled out. Okay. A fresh standard was also prepared and verified, and no cause was identified. Now, phase 2A, no cause was identified. The full-scale lab investigation was done. Hypothesis was planned. Container to container variation was planned by two analysts, one from the same container and the other from new container. Results were well within specification. Now, post hypothesis, the analyst started telling he had done one additional actuation. That is the reason there was an OS. Now, you know what happens when after phase 2, you try to talk to the analyst and he comes out with some different theory on what he has done. That means you will lose trust on all the initial work, right? Already analyst had mentioned that in the checklist we mentioned everything is done as per STP, everything is documented. This, like, you lose trust there. So potential challenges: fresh standards are prepared without appropriate rationale. Why are we preparing fresh standard? Hypothesis is not clear. Okay. OS did not reoccur. The analyst now mentioned that he had done additional actuation, no study. Then again, we have the hypothesis was not planned for doing that additional actuation and actually seeing that you will get an OS. Okay, so this is again flawed hypothesis. These are some examples. So I'll not go through more examples, otherwise we'll run out of time, I think. Right? We have 10 minutes. But you all can reach out to me again. That's not a problem, and we can have more discussion, because I have many examples about various types of OS. These are actual taken from the uh, uh, observations also. No root cause and still the manufacturing continued. So this is also important. Like, if you keep getting OS failures, can your manufacturing be continued? Okay, now I'll take, this is from an FDA presentation. Now, there was a case where duplicate samples of a product were tested for the assay. Duplicate sample results were 89.5 and 90.8. Average results were 90.2. Your specification was 90.0 and 110. Now, your phase 1 was no attributable error. Phase 2 retesting results are 91 and 91.4. Average is 91. Product released. Is this acceptable? That's the question now. What do you think? As a QA, will you release the batch or will you not release the batch? See the borderline cases. Okay, you will have to do many things. You have to check the history, the CPV, how's the trend, why it is at 91, what were the assay results during in process, whether anything happened, any losses happened. Okay, everything. How's the stability of the batch? Then case 2 is phase 1 investigation. There was an anomalous peak observed. So there was root cause was glassware contamination, not fully dried. Hypothesis is that anomalous peak is residual cleaning solvent. Phase 2, retesting by second analyst, and anomalous peak disappeared. Product is released based on retest. So you have to think now, what is missing in this investigation? Okay. They just said the peak is a residual cleaning solvent because it was not fully dried. We'll have to check if the same method of cleaning is used for all other cleaning, whether past what we have observed, what type of have we observed such type of peaks, what is this peak, unknown peak. You'll have to identify that also, because this becomes an extraneous type of peak also if it is from residual cleaning solvent. Okay. So these are the examples. Like, it is vital for the doer. Okay, this is, I have summarized all the examples over here to notify the anomaly at first instance, as it will always be beneficial. As I'm telling you, it is very important. The first hand information and the review is very, very important. Equally important is the role of supervisors who receive the first hand information, and the first ones to review the observation goes to the bench and sees what has happened. Supervisor should be aware of all the critical steps of the method and should be able to ask the right question. As we saw some of the examples here, OS during ointment preparation, missed to identify, okay, what was missed to identify differences, boiling water bath or boiling at heating at 60 degrees, shaking, whether you require. Then during short wait, OS of the short wait of nasal pump, So whether you have to shake or not shake. Then during dissolution, whether the many times, in dissolution you will see, if you pick up the 483s and read, all the dissolution investigation is done, and then they write a cause that I maybe the paddle was not attached. But you always have a checklist when you start the analysis, right? By the supervisor, that paddles are attached, tablets are dropped, everything is done for the dissolution. And towards the then section 2, phase 2, we say okay, maybe the paddle is not attached. So that will break the trust of the investigators reading the investigation. So now comes the most important thing, is documentation. So you have done all the good work of investigation and then you don't document. What will happen? After 2 years, 3 years, you are gone. No one is there. Like the people keep changing, right? And when the investigators come, what they will find is written. Who will understand? You have to understand your audience. Who is the audience? Your regulatory inspectors, your QA, okay, your clients. They are your audience. So you have one chance to document everything very clearly, because it is a document that will talk to the auditors. You will not be available, because if you don't write a good story, okay, no one will understand what has happened. Like I'll give you an example. There was a very simple bulk density tap density investigation, OS. Okay, no assignable cause was found in phase 1 part A. Suspect cause was identified as incorrect glassware used for analysis. Analyst did not use class A glassware cylinder. Retest was performed using class A. Original results were invalidated. Finished. But still we got cited for this observation. Okay, this was the original OS we had done. The gaps, what the investigator auditor told us, did not discuss the method requires to use, whether your method says you have to use class A cylinder for tap density. That was not acknowledged, the gap in the procedure, and gap identified was not extended how the past batches were tested, whether they were tested with class B or class A. So though this was a very simple investigation, still it got cited in the 483. Okay, phase A 1A should have identified the cause there only, but it was not mentioned that everything is good and everything was done properly. Discussion on standard accessories should have to be used for bulk density and tap density should have been done in investigation. It was not done. Rationale why phase 2A is not required should also be written in your investigations. Okay, gap in method was noted, but there was no action, like whether we have to review other methods, whether we have to review the past batches. Then impact of this method on past analysis was not done. And then as a result, like a big observation was cited and the company had to give justification and give clarification for the observation. Okay, those were the consequences. So final thoughts: OS investigations are time consuming but very important and key. Why? Because the product is in the market and patient safety is at risk. Always remember that. On robust OS investigation, OS SOP is very critical, very critical. Every audit OS and OS SOP gets reviewed. Okay, need for employee training is equally important for supervisors also. Investigation plans must be predefined. It should not be randomly you keep doing some investigations. Then suspect cause should be studied with experiments. Multiple departments may be involved. It is not that it is only the duty of QC to do the investigation or QA. Okay, you should have a cross-functional team when it's a full-scale investigation to be done. Many companies have started having cross-functional dedicated teams. Microbiologists, chemical, manufacturing, QA, QC sitting together, forming a team from manufacturing, science, technology transfer person, and then doing the brainstorming and doing all the investigation. Scientific rationale needs to be used at each step. Rationalization is very important. Why I'm doing this? What is the outcome? How I'm going to interpret my results? QA needs all the data. Everything has to be submitted. Nothing can be discarded. Okay, and CAPAs have to be implemented and effectiveness checks verified. Testing into compliance is BUAO, like it's not correct. Potential significant consequences for improper investigations, everyone knows, like there's your sites may get potential warning letters. And most important but not the least is documentation. Appropriate documentation is very important. Okay, so with that we complete the session. Thank you very much.
Okay, thank you so much, ma'am. We'll take
Hello.
some questions.
Yes.
Yes, ma'am. Take some questions. There are a few in the
Yes.
chat box.
Yes.
Can you see the chat box, ma'am?
1 minute.
I'll read if you want.
Yes,
There are some.
please read. No problem.
Okay, Pradipti asks that she teaches pharmaceutics in for students. So,
Okay.
what is one thing about OS root cause analysis that is never written in the textbooks, but actually happens in the industry? How can we better prepare students to handle such high pressure environment for a QC failure?
Yeah, you need to have what I think is, Dipti, that's a very good question. And, there's a gap, I think, when people come from college to industry about investigation. There needs to, you need to have guest lectures, and you need to have some, uh, workshops on how people will write investigations. That will help in colleges, especially. That's the question, right? How that gap has to be bridged.
Yes, ma'am. Correct.
Yeah, that's how I think, you should have more guest lectures and have various workshops so that people get trained. And you very well said that, yes, there's a gap in industry and what we learn in college. Even when I was in college and post graduation and all these things, when like not taught at all. So it's a very good thing that during your annual workshops or quarterly workshops, these topics, industry topics, are taught to the students so that they're well prepared when they go to the industry.
Correct. She also is asking that she prepares for institutional audits in the corporate world. When an OS is concluded, how is that knowledge archived to prevent recurrence?
That knowledge is through CAPA. Okay. So if you see 820.90 for medical devices, it clearly says that the CAPA information has to be disseminated, and then there are CAPA trends which are made. Okay. So many companies have this procedure when they're training their analyst on job also. So they have modules for various types of causes and kappas which they have implemented. So it's the through training only you can implement these things, that see do what are the do's and what are the don'ts in the investigation. And also quarterly and annual trends are there. So those trends are also, people get trained on those trends annually and quarterly. Many companies have their procedure that on annual trends, there's a training taken, cross-functional team attends that, like what are the types of issues we had identified, what kappas are taken. So this is a learning for everyone. That's how you have to share your learnings. Otherwise, it will remain in that investigation and close, and people will keep repeating. So that's why, in investigation, one key part is the past analysis. You have to see what are the kappas taken for the past investigations and how the trend was. Okay.
Absolutely correct. Another question is that, ma'am, is there a shift from reactive OS to predictive OS using machine learning? How can we, as researchers, incorporate these industrial predictive models into our initial formulation or evaluation stages?
So yeah, that's a very good question. Yes, that is happening now. People have started using, provided you know how they're doing it. You have a golden batch concept, like all the in process parameters were good, the results were good, the stability was good, and then compare the other batches. That's called as ongoing monitoring. Okay, then each batch when it does, you have to do an ongoing monitoring, and then through predictive analysis you will know whether this batch will meet the specification, whether it's an out of trend today, and whether it will meet the specification or no. Through statistical evaluation you could do that predictive analysis.
Okay, ma'am. Thank you. Then,
There is one question.
yes, Ravi. Yes,
How
yes,
do
please.
you handle OS in a continuous manufacturing system compared to batch processes? In manufacturing
Sorry,
and process,
I didn't continuous
continuous manufacturing systems compared to batch processes.
compared to batch processes means, like, I didn't get your point.
There is one system is continuous manufacturing, right from
Correct,
start to
correct.
the end. and another is batch processes. So in continuous manufacturing system, how do you handle OOS?
It's the same way. OS handling is the same. It's not that for continuous manufacturing it is different or batch processing it is different. If it is a continuous manufacturing process, okay, and you have seen an OS observation, it's not that you're not going to do the test for continuous manufacturing. You will have to do it, right? For continuous manufacturing also. Unless you have included some reduced testing program and you are doing a skip testing and you are not doing some test in continuous manufacturing. So it depends that. So there's no difference in handling OS in batch processing or continuous manufacturing.
Okay, thank you.
I would like to add a small point here. When it is continuous manufacturing, the basics of OOS does not change, but you need to have online tools so that you don't keep on producing OOS material. That means, for example, in a water system you have online TOC. If it detects a out of specification, it should cut off the supply and your production of WFI or something should stop. Otherwise, you will keep on producing non-standard material.
Yeah, so that's a requirement, or during qualification itself, you have to identify all those things. Yeah.
Correct. Standard. Okay, another question. Standard OS investigations often focus on simple assays or dissolution. For complex systems like liposomes, where critical quality attributes include other parameters, how does the industry adapt OS root cause analysis when the variability might be inherent to the nanostructures rather than an analytical error? So she's asking that
So
the
I'm
formulation
not
is
an expert
complicated.
in liposomes though, but you know what? The guidance already says that, you know, if your method itself has inherent error, inherent variation, that should be seen in your validation and you have to define it in your method itself. Okay, you will be doing so many replicates and among the replicates, this is my variation accepted. That's what. If anyone else has any other question on liposome, like anyone can answer.
That was the question, ma'am. Not with specific to liposome. She was just asking that the product itself has a inherent problem. The answer was the question was with respect to that only. That if the product has an inherent error, what should we be doing? Okay.
So one more question is there. Should
Yes,
AI be used in OS trend detection? Discuss benefit and risk of that.
AI be used in trend analysis. Yes, you could
OS
use
trend detection.
AI.
Can be used.
You have a system.
Benefit and
Yes,
risk.
yes, you could use AI. It's not that you cannot use AI provided someone reviews what the results are. See, blindly, very recently, like this week, FDA has given a warning letter to a company who had used AI to do the specification, but it was not reviewed. Okay. So it's not that totally you have to rely on AI and then say, okay, AI has done. Someone has to, the QA has to review QC what all things AI is generating, whether it's acceptable.
But if AI passes their international standard specifications, then it can be.
Then yes, if it's a hypothetical thing, yes.
There's a 27,000 standard is there of AI. 20,000.
Whatever, yeah. If you think you're confident, yes, why not? The world is moving towards AI. There's no one has stopped you that you cannot do that, right?
Only thing is to justify.
Yes, it has to be justified, because when I also use AI in many things and tell them to summarize and then myself read the original document, there are so many errors which they give out, the summary. So you have to be very careful. Okay. Everyone is jumping into AI now. You have to be careful. Yeah,
I
that's
think
what
the point that was put forward in that warning letter also was that there was never it was mentioned that you shouldn't be using. It was mentioned that you can use, but you need to review, and the ultimate responsibility should be with the person.
100%.
Okay, ma'am. Another question, if I can add one more. when is averaging considered inappropriate in QC results, even if numbers look close?
When is averaging acceptable? Averaging is not
When is
acceptable.
when is averaging considered inappropriate, not proper, not appropriate?
So it is never appropriate. So it's close or not close. That's what FDA says.
Okay, madam. I guess what is okay. That okay. Yuvraj asks, what is FDA view on human error? Does training considered is training is considered as a CAPA?
No. So that is one of the warning letter and 483 observation. If you do an OS and just say this is the training I'm giving, and you keep repeating that, okay, out of your 100 investigations closed, and 50% you're telling human training as the CAPA, only CAPA, training as only CAPA. So you will surely get a 483 for that. So you cannot have human error training as only CAPA. No, that's acceptable. Once a while, you may be it may be acceptable, but if you see your trend analysis, that should be one of your quality management review parameters also, that how many investigation I have closed, and from these closed investigation, what are the investigation which mention only training as the CAPA, and then appropriate actions have to be taken. Why I am doing that? By training. Okay. So there was, I can see some comments. People are saying that voice was not clear. I'm sorry for that, but I'm not sure.
No, that was okay. It was related.
I think I'll have to modify my speaker and audio. Yeah.
Okay. Yes, we will be sharing the videos. So there are few questions in the last, ma'am. Can you see that? One,
Sure.
two, you
Yeah.
have already replied. One last from John.
I have a question specifically in phase 2, correct? Should I always open a deviation when entering phase 2 or only when the root cause is identified? No, you have to see phase. It's not that open a deviation. Depends on how you have written your procedure. So phase 1, it goes that way. Phase 1 and phase 2 investigation. Phase 1 is lab. You did not identify. It goes to phase 2 laboratory manufacturing investigation. So maybe your procedure says you open a deviation. But generally, what other companies, normal industry does is it's part of that OS investigation itself. They don't open a separate deviation for that. One branch, if you have a trackwise will open a child for the manufacturing investigation, and that's how it is done. Okay. I would also like you to clarify phase 3. Whenever phase 2 is initiated, is it mandatory? No. So whenever phase 2 is initiated, it is not mandatory to do phase 3. No, that's not the concept. Phase 2, if your root cause investigation does not identify any lab error or manufacturing and your results kept failing, you have to do your batch disposition, close the investigation. That's how phase 2 closes, and then initiate phase 3. Phase 3 can be done only once batch disposition decision has been taken. Okay. So next question will go up. During OS investigation, how do we systematically identify the root cause, especially when multiple factors like, so this is again, I'm telling you, there's the same question Sanjana has asked. During OS investigation, what generally I have seen is doing the fishbone analysis, 6M method you call it. Okay, that's a systematic way of determining the root cause. First is the lab, do the first interview, the obvious error, and then go to the phase 1B remeasurement, rationalize each step why you want to do, then go to the 6M, what is the sampling procedure, what is the method, what is my standard, whether how my AMB was written, whether any changes in STP, all those things. They're the systematic, like, all the factors you need to do, and it should be well documented in your procedure. Okay. Then is Pradipti. She's telling in synergistic co-delivery system, we often see a dependency in stability, correct? If one drug stays within spec, but the second drug, thymoquinone, falls out of, does the industry typically revalidate the entire synergistic ratio, or it is the investing limited to the single failed component? So it is generally to that single. If you're telling, what I understand is your multi-product, right? In the single injection, both drugs, one drug was showing OS and other was showing not OS, passing. So of course your investigation you will have to do for the sample, and when you are doing the remeasurement, and for that passing, originally passing is kept on giving the same result, then it's fine. But even then that starts behaving in a different manner, then that's a challenge for you. How does data integrity impact OS investigation? Sanjana is asking. So Sanjana, this is very important. Both are interrelated. OS investigation impacts data integrity when you discard your results and you do not report all the results and you choose only passing results. FDA will tell this is a data integrity issue. Okay. And you keep your analyst without you do unauthorized testing. That is data integrity issue. Trending of results are mandatory and every quarter training should be given to quality. Yes. So Ramesh is telling, what is FDA view on human error? Does training? So we have answered this question. I think we have taken the questions now, Sujit.
Yes, ma'am. The some of them are repetitive. So,
no, I took it from down. You said down, yeah.
Yes. You have finished. Yeah, all the questions have been done.
Thank you very much.
Yes, ma'am. Thank you so much, ma'am, for your efforts. It was such a great session.
Yes.
We dealt with every topic. Means we can. I guess most of them can start.
Because 1 hour, 40 minutes. You said it's less because if you take examples, I have many examples based on the live studies.
But you did justice to the topic, ma'am. Thank you so much, and for
thank you. I also enjoyed it. Thank you very much, everyone.
Thank you everyone for staying the course, and we'll see you in the next session. Do definitely give us your reviews regarding the session.
Please give the review on LinkedIn also. Thank you so much.
Yes, ma'am. Thank you. And also with respect to the certificates, please ping me with your IDs. I'll definitely issue the certificates. Thank you so much.
Sure.
Happy weekend.
Thanks, Bye. Have a good evening. Bye, everyone.